Welcome to our channel. We are so glad you found your way here. Let us help you fall asleep. Let's begin with the endosymbiotic theory, when two cells became one. Take a slow, deep breath in and release it. Let the weight of the day fade away as we embark on an unhurried, atmospheric journey through the wonders of the cosmos. Your mind is quiet, the universe is vast, and we are ready to explore. This theory posits that certain organelles within eukaryotic cells, such as mitochondria and chloroplasts, originated as free-living prokaryotic organisms. Over time, these entities formed symbiotic relationships with ancestral eukaryotic cells, leading to a merger that resulted in a more complex form of life. This union allowed for greater efficiency and adaptability, marking a pivotal moment in the evolution of life on Earth. The significance of the endosymbiotic theory lies in its profound implications for understanding evolutionary biology. It challenges traditional views of organismal evolution by suggesting that cooperation, rather than competition, can be a driving force in the development of complex life forms. This theory provides insight into the origins of cellular structures that are essential for processes such as respiration and photosynthesis, underscoring the intricate interplay between different life forms. Evidence supporting this theory includes genetic data revealing similarities between the DNA of mitochondria and that of certain bacteria. Additionally, the structures of these organelles closely resemble prokaryotic cells. Such findings reinforce the notion that the evolutionary history of life is not solely a linear progression, but rather a complex network of interactions and integrations. This perspective opens new avenues for research, inviting deeper exploration into how life on Earth has been shaped through symbiotic relationships over billions of years. Early theories of cellular origins often reflected a limited understanding of the complexity inherent in biological systems. The notion of spontaneous generation, prevalent from ancient times until the 19th century, suggested that life could arise from non-living matter. This idea endured until the experiments of Louis Pasteur demonstrated that microorganisms came from existing life. Such revelations paved the way for a more nuanced appreciation of cellular development. The dawn of the microscope in the 17th century unveiled a hidden world, revealing that life was not confined to visible organisms. Pioneers like Antony van Leeuwenhoek observed single-celled entities, which sparked curiosity about their origins. The concept of the cell as the fundamental unit of life gained traction in the mid-19th century, with the formulation of the cell theory by Schleiden and Schwann. This framework positioned cells as distinct entities, yet it struggled to explain the origins of organelles within them. The gradual acceptance of the nucleus and mitochondria as vital components of cellular function drew attention to the need for a comprehensive understanding of their genesis. This inquiry led scientists to contemplate the possibility that these organelles might have originated from free-living prokaryotic organisms, setting the stage for the development of the endosymbiotic theory, an explanation that would redefine the narrative of evolutionary biology. Symbiosis describes a close and often long-term interaction between different biological species. This relationship can take various forms, each characterized by differing degrees of benefit or harm to the organisms involved. Mutualism represents one of the most well-known types, where both species derive benefit from the interaction. An example can be seen in the relationship between pollinating insects and flowering plants. The insects obtain nourishment from nectar while facilitating the plant's reproduction. Commensalism, another form, indicates a relationship where one organism benefits while the other remains unaffected. This can be observed in barnacles that attach themselves to a whale's skin, gaining mobility and access to nutrient-rich waters without harming the whale. Parasitism, conversely, highlights a relationship that is detrimental to one party. In this case, the parasite benefits at the expense of the host, as seen in various organisms that live within or on a host organism, extracting nutrients and often causing harm. The concept of symbiosis extends beyond ecological relationships and into cellular interactions, particularly within the realm of endosymbiotic theory. The fusion of distinct organisms can lead to significant biochemical advancements, enabling complex life forms to thrive. This intricate interplay underscores the importance of cooperation and interdependence in biological systems, influencing the evolutionary trajectory of life itself. The origins of life trace back to a time when the planet was an inhospitable environment, characterized by volcanic activity and a dense atmosphere rich in gases. This primordial landscape offered a unique mixture of chemical compounds, which provided the building blocks for the first organic molecules. The presence of water, in its various forms, played a critical role in the formation of these essential components. In this region, conditions favored the synthesis of amino acids and nucleotides, the fundamental units of proteins and genetic material. Over time, these molecules began to organize, forming simple structures that displayed properties of self-replication and metabolism. Such early cellular entities were not yet true cells, but exhibited characteristics that hinted at the complexity to come. The emergence of lipid membranes marked a pivotal moment, allowing these molecular assemblies to encapsulate their environments. This led to the formation of protocells, which were capable of maintaining an internal environment distinct from their surroundings. Within these structures, rudimentary metabolic processes could occur, paving the way for a more complex organization. In this evolving landscape, chemical gradients and energy sources facilitated interactions between different molecular inhabitants. Such interactions laid the groundwork for more sophisticated cellular development, leading eventually to the diverse life forms that would dominate the planet in the ensuing epochs. Pathologia cells represent a fundamental stage in the evolution of life, characterized by their simplicity and efficiency. These microscopic entities lack a nucleus, a varsis volmostic, with their genetic material dispersed within the cytoplasm, allowing for rapid reproduction and adaptation to a variety of environments. The absence of membrane-bound organelles further distinguishes them, showcasing a streamlined structure that has persisted for billions of years. In this evolutionary journey, prokaryotic cells have demonstrated remarkable versatility. They occupy diverse habitats, from deep-sea vents to arid deserts, exhibiting an extraordinary ability to thrive under extreme conditions. Essential processes such as photosynthesis and nitrogen fixation can occur within these cells, highlighting their role in maintaining ecological balance. The evolutionary history of prokaryotes is marked by significant events, including horizontal gene transfer. This process facilitates genetic exchange between different species, accelerating adaptation and innovation. Such mechanisms have been crucial in shaping the genetic diversity observed in various prokaryotic lineages today. These cells also serve as the evolutionary precursors to eukaryotic organisms. The endosymbiotic events that eventually led to the emergence of more complex life forms stemmed from ancestral prokaryotic interactions. As these interactions evolved, they laid the foundation for the intricate cellular systems that characterize eukaryotic life, showcasing the profound impact of prokaryotic cells on the tree of life. Mitochondria serve as the powerhouse of eukaryotic cells, playing a critical role in energy production. These organelles are descendants of prokaryotic cells, having been enveloped by ancestral eukaryotic hosts through endosymbiosis. This intimate relationship allowed for the development of a sophisticated mechanism known as oxidative phosphorylation, which generates adenosine triphosphate, or ATP. the primary energy currency of living organisms. Within the confines of these double-membraned structures, numerous biochemical reactions occur, converting nutrients into usable energy. The mitochondrial matrix houses enzymes essential for the Krebs cycle, a pivotal process in cellular respiration. This region operates under strict regulatory mechanisms that ensure efficiency and adaptability to the cell's metabolic demands. Mitochondria also possess their own circular DNA, resembling that of bacteria, a remnant of their evolutionary origins. This genetic material encodes vital proteins necessary for mitochondrial function, further emphasizing their dual heritage. They are involved in additional cellular processes, including apoptosis, which is a form of programmed cell death crucial for maintaining cellular homeostasis. The dynamic nature of mitochondria allows them to adapt to changing environmental conditions, influencing their morphology and distribution within the cell. This adaptability underscores the essential role these organelles play in the survival and functionality of eukaryotic organisms, marking them as central players in the evolutionary narrative of life on Earth. The role of chloroplasts in photosynthetic cells epitomizes the fascinating interplay between light and life. These organelles, found predominantly in plant cells, are responsible for capturing sunlight and converting it into chemical energy through the process of photosynthesis. Within their membranes, chlorophyll molecules absorb specific wavelengths of light, primarily in the blue and red regions of the spectrum. This absorption initiates a series of biochemical reactions, transforming carbon dioxide and water into glucose and oxygen. The intricate structure of chloroplasts enhances their efficiency. Thylakoid membranes, organized into stacks known as grana, provide ample surface area for the light-dependent reactions. This compartmentalization allows for a highly efficient transfer of energy, as the captured photons drive the synthesis of adenosine triphosphate, or ATP, and nicotinamide adenine dinucleotide phosphate, or NADPH. The subsequent light-independent reactions occurring in the stroma utilize these energy carriers to fix carbon dioxide into organic molecules. Beyond their immediate role in energy conversion, chloroplasts also contribute to broader ecological systems. They facilitate the oxygenation of the atmosphere and serve as the foundation of food web's path, sustaining diverse organisms. This interdependence illustrates how chloroplasts are not merely cellular structures, but vital components of Earth's biosphere. Key figures have shaped the understanding of the endosymbiotic theory. Lynn Margulis stands at the forefront. Her research challenged traditional views of evolution, emphasizing symbiosis as a driving force. She proposed that eukaryotic cells originated from the merging of distinct prokaryotic organisms. Her theories faced skepticism, yet over time, accumulating evidence bolstered her claims, reshaping the scientific narrative. Margulis's work, particularly in the 1960s and 70s, highlighted the importance of cooperation among microorganisms. This perspective shifted attention from competition to collaboration as a fundamental aspect of evolution. The ramifications extended beyond cellular biology, influencing broader ecological and evolutionary thought. Colleagues and contemporaries contributed to this evolving dialogue. Researchers like Carl Woese advanced the understanding of microbial diversity, revealing the complexity of life at the smallest scales. Their methodologies, combining molecular biology and genetic analysis, provided tools to validate Margulis's hypotheses. The discovery of similarities between chloroplasts and cyanobacteria, as well as mitochondria and proteobacteria, reinforced the concept of symbiotic origins. Key findings in molecular genetics have illuminated the intricate relationships that define cellular life. The resulting synthesis of knowledge demonstrates that cellular evolution is not just a tale of survival, but a nuanced interplay of life forms, fostering resilience and adaptability in the biosphere. This intricate relationship is further illuminated by morphological evidence, particularly structural similarities between organelles and prokaryotic cells. Mitochondria and chloroplasts, for example, exhibit distinct features reminiscent of bacterial cells. Both organelles possess a double membrane structure, resembling the characteristics of prokaryotic cells, which typically exhibit simpler membranes. The inner membranes of these organelles are intricately folded, providing an increased surface area for metabolic processes, akin to the folding seen in the membranes of certain bacteria. This configuration supports the notion that these organelles, through evolutionary history, originated from free-living prokaryotes that entered a symbiotic relationship with ancestral eukaryotic cells. Additionally, the size of mitochondria and chloroplasts parallels that of bacteria, further reinforcing this connection. The ribosomes present within these organelles share similarities with those found in prokaryotes, differing significantly from eukaryotic ribosomes in both size and structure. This observation provides insight into the evolutionary process, suggesting that these organelles retain an ancient lineage linked to their prokaryotic ancestors. Genetic analysis reveals that the DNA within mitochondria and chloroplasts is circular, much like bacterial DNA, contrasting with the linear DNA found in the nucleus of eukaryotic cells. These morphological and genetic similarities form a compelling foundation for the endosymbiotic theory, highlighting the complexities of cellular evolution and the interdependence of life forms. Mitochondrial DNA provides critical insights into the evolutionary lineage of eukaryotic cells. This genetic material, distinct from the nuclear DNA housed within the cell's nucleus, exhibits significant prokaryotic characteristics. The circular structure of mitochondrial DNA mirrors that of bacterial genomes, suggesting a shared ancestry. This observation supports the hypothesis that mitochondria originated from an ancestral prokaryotic organism. Genetic sequencing reveals that mitochondrial DNA is more closely related to certain groups of proteobacteria than to the nuclear DNA of the host cell. This similarity extends to the sequences of genes involved in energy metabolism, which are fundamental to the mitochondrian's role in cellular respiration. Such findings illustrate not only the functional integration of these organelles, but also the historical merger that may have taken place billions of years ago. The inheritance pattern of mitochondrial DNA further strengthens this theory. Unlike nuclear DNA, which is inherited from both parents, mitochondrial DNA is passed exclusively from mother to offspring. This uniparental inheritance allows for tracing lineage and provides a clearer picture of evolutionary processes over time. These genetic connections underscore the dynamic relationship between prokaryotes and eukaryotes, emphasizing the importance of symbiotic interactions in shaping the complexity of life on Earth. Each piece of evidence, meticulously gathered, contributes to a comprehensive understanding of our planet's biological history. Ribosomal RNA comparisons provide profound insights into the evolutionary relationships among various life forms. The structure of ribosomal RNA, essential for protein synthesis, remains remarkably conserved across different species. This stability allows for the examination of genetic sequences, revealing connections that span billions of years. By analyzing the ribosomal RNA sequences, scientists can trace the lineage of organisms, identifying common ancestors and divergence points within the tree of life. In this region of research, the similarities and differences in ribosomal RNA sequences serve as markers of evolutionary change. Variations reflect adaptations and speciation events, highlighting how life has responded to environmental pressures over eons. These molecular comparisons facilitate the reconstruction of evolutionary pathways, offering a clearer picture of how distinct groups of organisms emerged. Notably, when examining the ribosomal RNA of eukaryotes and prokaryotes, profound insights into their relationships surface. The endosymbiotic theory posits that certain organelles within eukaryotic cells originated from prokaryotic organisms, suggesting a pivotal moment in evolutionary history. The ribosomal RNA evidence supports this theory, indicating a shared ancestry that underpins the complexity of life as we know it. Understanding these relationships sheds light on the mechanisms of evolution, demonstrating the intricate web of connections that unite all living beings on Earth. Each discovery adds depth to the narrative of life's rich diversity. The role of endosymbiosis in evolutionary biology is pivotal, highlighting how complex life emerged from simpler organisms. Genetic material from once independent prokaryotic cells has been absorbed and integrated into eukaryotic cells, facilitating significant evolutionary advancements. This integration led to the development of organelles, such as mitochondria and chloroplasts, which are essential for energy production and photosynthesis. In this dynamic process, one organism lives within another, establishing a mutually beneficial relationship. Such interactions have not only influenced cellular function, but have also driven speciation and adaptation across various environments. The genetic exchanges among these organisms contribute to the overall genetic diversity observed today. Research continues to unveil the complexities of these relationships, revealing how endosymbiotic events have shaped the evolutionary tree. Mitochondrial DNA, for instance, shows similarities to that of certain bacteria, indicating a shared ancestry that dates back millions of years. The presence of chloroplasts in plant cells reflects a similar narrative, revealing a history of cooperation and adaptation. The implications extend beyond individual species, offering insights into ecosystem dynamics and the interdependence of life forms. This ongoing exploration of endosymbiosis serves as a lens through which the intricate connections and evolutionary pathways of life can be understood, further enriching the scientific discourse surrounding life's origins and diversity. The timeline of eukaryotic evolution reveals a gradual transition from simple prokaryotic organisms to the complex cells that define much of life today. Roughly 2 billion years ago, ancestral prokaryotes began to engage in symbiotic relationships, leading to the integration of distinct cellular components. This pivotal moment marked the beginning of eukaryogenesis, where the merger of different lineages gave rise to cells with a nucleus and membrane-bound organelles. The endosymbiotic events that followed are crucial for understanding this transformation. Mitochondria and chloroplasts, essential for energy production and photosynthesis, respectively, originated from free-living bacteria that entered into a mutualistic relationship with host cells. Over time, these engulfed organisms lost their independence, evolving into integral parts of the eukaryotic cell. Subsequent diversification occurred over hundreds of millions of years, leading to the emergence of various eukaryotic lineages. Protists, fungi, plants, and animals branched out, each adapting to their environments and developing unique features. Genetic evidence supports these evolutionary transitions, showcasing the shared ancestry among diverse life forms. The interplay of genetic exchange and environmental pressures contributed to the complexity seen in contemporary ecosystems. Understanding this timeline illuminates the profound interconnectedness of all living organisms, offering insights into the processes that shaped life on Earth. The evolution of the nucleus marks a significant milestone in the history of cellular development. This organelle serves as a control center, harboring genetic material and regulating cellular functions. Within these confines, eukaryotic cells gained the ability to manage complex processes, allowing for greater specialization and adaptability in response to environmental changes. In the context of endosymbiosis, the presence of a nucleus is crucial. It creates an environment conducive to the integration of distinct cellular identities. Organelles, such as mitochondria and chloroplasts, evolved from once free-living prokaryotes, entering host cells and forming a mutually beneficial relationship. The nucleus facilitates the coordination of these interactions, ensuring that the symbiotic entities contribute to the overall functionality of the host organism. These relationships exemplify how the nucleus not only protects genetic information, but also orchestrates the intricate processes necessary for cooperation between various cellular components. As eukaryotic cells evolved, the nucleus became a hub for genetic exchange and innovation, fostering diversity among life forms. The dynamics within this cellular environment illustrate the complexity of evolutionary processes, emphasizing that the emergence of the nucleus played a pivotal role in shaping life as it is known today. This connection between endosymbiosis and nuclear evolution underscores the remarkable journey of life on Earth. The emergence of oxygen transformed the early atmospheric conditions of Earth, playing a pivotal role in the development of eukaryotic cells. This gas, initially a byproduct of photosynthesis performed by cyanobacteria, steadily accumulated in the atmosphere, leading to significant biochemical shifts. As oxygen levels increased, environments became more oxidative, promoting the evolution of complex cellular structures. Metabolic pathways adapted to utilize oxygen, enhancing energy production through aerobic respiration. This process permitted cells to extract greater energy from organic materials, supporting larger and more complex organisms. The transition from anaerobic to aerobic environments facilitated the development of intricate cellular machinery, allowing for specialization and division of labor within cells. Such an environment fostered the symbiotic relationships necessary for eukaryotic evolution. The engulfment of anaerobic prokaryotes by larger cells, which had begun to utilize oxygen, led to the establishment of mitochondria. These organelles became essential for energy production, providing the fuel required for the burgeoning complexity of life. The interplay between rising oxygen levels and cellular evolution highlights a fundamental shift in Earth's biosphere. This gradual accumulation of oxygen not only reshaped atmospheric conditions but also catalyzed the intricate web of life that characterizes the planet today. Each step in this process reflects a finely tuned response to environmental changes, underscoring the interconnectedness of life and its surroundings. The endosymbiotic origin of mitochondria provides a compelling case study in evolutionary biology. This particular organelle, recognized for its role in energy production, is believed to have originated from a free-living prokaryotic ancestor. Over time, a primitive eukaryotic cell engulfed this bacterium, leading to an unprecedented level of cooperation. The engulfed organism gradually evolved into a permanent resident, facilitating a profound transformation in cellular function. Intriguingly, this fusion allowed the host cell to exploit the bacterium's efficient aerobic respiration capabilities. The energy derived from oxygen-based metabolism significantly enhanced the host's survival prospects in a changing environment. As generations passed, the integration deepened, with the engulfed bacterium transferring a substantial portion of its genetic material to the host's nuclear genome. This genetic exchange facilitated the establishment of a complex regulatory relationship between the two entities. Mitochondria began to lose their independence, retaining only the genes essential for their specialized role. Their double membrane structure serves as a vestige of their ancestral lineage, while the unique DNA they possess bears striking similarities to that of certain modern prokaryotes. The evolution of mitochondria exemplifies the intricate dance of cooperation and adaptation that characterizes life's history, illustrating how symbiotic relationships can lead to profound evolutionary innovations. The endosymbiotic origin of chloroplasts represents another pivotal chapter in the narrative of cellular evolution. This process began when a eukaryotic ancestor engulfed a photosynthetic cyanobacterium. Initially, this event appeared to be a straightforward act of predation, yet it transformed into a lasting partnership. Over time, the engulfed cyanobacterium relinquished its independence, becoming a permanent resident within the host cell. The relationship blossomed into a mutualistic symbiosis, as the cyanobacterium provided the host with the ability to convert sunlight into energy through photosynthesis. This new capability allowed the host cell to harness solar energy, fundamentally altering its metabolic processes. The engulfed organism gradually lost its original genome, transferring many essential genes to the host's nuclear DNA. This genomic integration facilitated the coordination of cellular functions, refining the efficiency of the photosynthetic apparatus. Chloroplasts, now integral to plant cells, exhibit distinct genetic characteristics that echo their prokaryotic origins. Their double membrane structure and ribosomal composition serve as reminders of their evolutionary lineage. This adaptation also enabled the emergence of vast forests and ecosystems, significantly influencing atmospheric composition and climate over geological timescales. The endosymbiotic events that produced chloroplasts not only reshaped individual cells but also catalyzed the evolution of life on Earth, fostering diversity and ecological complexity. The interplay of genetic material between different organisms plays a vital role in evolutionary processes. Horizontal gene transfer facilitates the exchange of genes across species, significantly impacting adaptation and survival. This mechanism allows for the rapid acquisition of beneficial traits, bypassing the slower pace of vertical inheritance. Microbes exemplify this process, often sharing genetic information through various methods such as transformation, transduction, and conjugation. This exchange can confer advantageous characteristics, such as antibiotic resistance or metabolic capabilities, enhancing their resilience in diverse environments. Such adaptations underscore the fluidity of genetic boundaries, illustrating that evolution is not solely a linear progression, but rather a complex web of interactions. In multicellular organisms, the implications of horizontal gene transfer are profound. It introduces new genetic variations that can influence development and physiology. For instance, some plants have incorporated genes from bacteria, enabling them to synthesize novel compounds or adapt to challenging conditions. This phenomenon demonstrates the interconnectedness of life forms, where genetic material transcends species barriers, leading to innovative evolutionary pathways. Research continues to unveil the intricate networks formed through these transfers, revealing an underlying unity among living organisms. The historical significance of gene transfer reinforces the notion that evolution is a collaborative endeavor, highlighting the shared genetic heritage that binds all life on Earth. Competing theories of eukaryotic origins contribute to the ongoing discourse surrounding life's complexities. Various hypotheses have emerged alongside endosymbiotic theory, each offering alternative explanations for the emergence of eukaryotic cells. The autogenous theory posits that eukaryotes arose from the gradual evolution of prokaryotic ancestors through internal modifications. This concept emphasizes self-organization within cellular structures, suggesting that complexity arose from an innate capacity for self-improvement. The symbiogenetic theory, while related, diverges by focusing on the symbiotic relationships between different prokaryotic organisms. It proposes that cooperative interactions led to the formation of new, complex life forms, This perspective highlights the importance of environmental factors and the ability of organisms to adapt through mutualistic relationships. Another consideration involves the possibility of genetic exchange between archaea and bacteria, which further complicates the understanding of eukaryotic lineage. This horizontal gene transfer presents a scenario where genetic material is shared across species, blurring the lines of evolutionary relationships. Each of these theories underscores the intricate web of interactions that may have led to the emergence of eukaryotes. The distinct pathways suggested by these hypotheses reflect a multifaceted evolutionary landscape, one characterized by cooperation, adaptation, and the dynamic interplay of life forms throughout Earth's history. Understanding these perspectives enriches the narrative of life's origins. Phylogenetics plays a crucial role in unraveling the intricate relationships between organisms, particularly in the context of endosymbiosis. By examining the genetic lineage of various species, scientists can trace back the evolutionary paths that led to the formation of complex cells. This investigative approach reveals how certain organelles paths that led to the formation of complex cells. This investigative approach reveals how certain organelles, such as mitochondria and chloroplasts, originated from free-living prokaryotic ancestors that were engulfed by ancestral eukaryotic cells. The genetic data obtained from contemporary organisms enables the reconstruction of evolutionary trees, illustrating the connections between disparate life forms. Each branch of these trees symbolizes a lineage that has diverged over millions of years, some leading to the development of multicellular organisms, while others remained as single-celled entities. The similarity in DNA sequences between these organelles and specific bacteria provides evidence of their shared ancestry, confirming the endosymbiotic events that shaped cellular evolution. In this region of scientific inquiry, the importance of horizontal gene transfer cannot be understated. This phenomenon, where genetic material is exchanged between different species, further complicates the understanding of evolutionary relationships. The implications of these insights extend beyond cellular biology, influencing perspectives on biodiversity and the adaptability of life. All these intricate connections underscore the importance of phylogenetics in elucidating the history of life on Earth, revealing the delicate interdependence that has characterized biological evolution. The endosymbiotic theory has reshaped the understanding of evolutionary trees, providing a framework to visualize the relationships among diverse life forms. It illustrates how certain organelles, such as mitochondria and chloroplasts, originated from free-living prokaryotes that entered into a symbiotic relationship with ancestral eukaryotic cells. This integration marks a pivotal moment in evolutionary history, leading to significant diversification. Phylogenetic analyses reveal that these organelles possess their own DNA, distinct from the nuclear genome, further supporting their prokaryotic origins. This genetic evidence allows scientists to trace lineage patterns, creating branches that reflect the shared ancestry of modern organisms. The implications extend beyond solitary species, illustrating a shared evolutionary trajectory that connects plants, animals, fungi, and protists. The incorporation of endosymbiotic events into evolutionary trees highlights the complex interplay of cooperation and competition that drives life's diversity. Instances of horizontal gene transfer also emerge as critical factors, blurring the lines between distinct evolutionary paths. Organisms can exchange genetic material across species, complicating traditional lineage models. This nuanced understanding emphasizes the fluidity of evolutionary processes, where cooperation can lead to profound transformations. The endosymbiotic theory thus serves as a cornerstone in evolutionary biology, prompting a re-evaluation of how life has adapted and thrived in an ever-changing environment. Symbiotic relationships represent a fundamental aspect of ecosystems, where diverse organisms interact and coexist in ways that enhance survival and reproduction. In this intricate web, mutualism exemplifies a relationship where both participants benefit, such as the interaction between bees and flowering plants. Bees pollinate flowers while obtaining nectar, ensuring the continuation of both species. Commensalism illustrates another facet, where one organism gains advantages without affecting the other. Epiphytic plants thrive on trees, drawing moisture and nutrients from the air, while their hosts remain largely unharmed. This arrangement highlights the subtle balance maintained within ecosystems, often unnoticed yet crucial for biodiversity. Parasitism, contrastingly, demonstrates a relationship where one organism benefits at the expense of another. Parasites can significantly impact their hosts, influencing population dynamics and species evolution. The presence of such relationships contributes to the complexity of ecological interactions, shaping communities over time. These interdependencies illustrate the interconnectedness of life, where the survival of one species often relies on the presence of others. Ecosystems, through these symbiotic interactions, maintain resilience against environmental changes and disturbances. The delicate balance inherent in these relationships underscores the importance of preserving biodiversity, as each organism plays a vital role in sustaining the intricate fabric of life on Earth. This interconnectedness extends to cellular metabolism, which evolved significantly due to endosymbiotic events. Mitochondria and chloroplasts, descendants of once free-living prokaryotes, serve as prime examples. Their integration into eukaryotic cells marked a pivotal shift in how energy is produced and utilized. Mitochondria, often referred to as the powerhouses of the cell, facilitate aerobic respiration, converting nutrients into adenosine triphosphate, the energy currency essential for cellular activities. Chloroplasts similarly transformed metabolic processes through photosynthesis, enabling organisms to harness solar energy. This capability led to the production of glucose from carbon dioxide and water, releasing oxygen as a byproduct. The emergence of these organelles not only enhanced energy efficiency, but also allowed for greater complexity within life form. This region of biological evolution reflects a refinement in metabolic pathways, enabling organisms to adapt to varying environmental conditions. The implications of this symbiotic relationship extend beyond individual cells, influencing ecosystems and biogeochemical cycles. Oxygen levels on Earth rose dramatically, reshaping atmospheric composition and paving the way for the proliferation of aerobic life forms. The metabolic innovations stemming from endosymbiosis underscore the profound interconnectedness of life, illustrating how cooperative relationships can catalyze significant evolutionary advancements. Each cellular transformation resonates through the generations, influencing the tapestry of biodiversity observed today. The process of photosynthesis emerged as a pivotal evolution in cellular life, originating within simple cyanobacteria. These organisms harnessed sunlight, converting it into chemical energy, thus laying the groundwork for future life forms. As these bacteria thrived in ancient oceans, they contributed to the gradual accumulation of oxygen in the atmosphere, an event that would reshape the planet's chemical landscape. This transformation did not occur in isolation. For Diosso, the endosymbiotic relationship formed when larger eukaryotic cells engulfed cyanobacteria, leading to the development of chloroplasts. The integration of these photosynthetic partners allowed cells to exploit sunlight more effectively, enhancing growth and survival in diverse environments. Chloroplasts became indispensable organelles, facilitating the conversion of light energy into sugars, fundamentally altering the energy dynamics of ecosystems. The legacy of this evolutionary milestone is evident in the vast array of photosynthetic organisms present today. From towering trees to microscopic phytoplankton, they all trace their lineage back to that initial symbiotic event. This intricate relationship exemplifies the profound interconnectedness of life, demonstrating how cooperation among disparate organisms can yield remarkable adaptations over time. Case studies of modern endosymbiotic relationships reveal fascinating interactions, particularly between protozoa and algae. Within these associations, certain protozoa have developed the ability to incorporate and utilize algae for photosynthesis. These partnerships exemplify a significant evolutionary strategy that enhances survival and adaptability in diverse environments. In a specific instance, the genus Euglena showcases a remarkable blend of characteristics. This organism possesses both chloroplasts, derived from engulfed green algae, and heterotrophic capabilities, allowing it to thrive in nutrient-poor conditions. The retention of algal chloroplasts enables Euglena to harness sunlight, converting it into energy while simultaneously absorbing organic material from its surroundings. Similarly, the marine protozoan Paramecium demonstrates a unique integration of algal symbionts. Through this symbiotic relationship, it acquires the ability to produce energy via photosynthesis, which supplements its diet. The algae not only provide essential nutrients, but also contribute to the overall fitness of Paramecium in its aquatic habitat. These examples illustrate how modern endosymbiotic relationships are not merely relics of evolutionary history, but active, dynamic systems that continue to shape the biology of organisms today. Such partnerships highlight the ongoing significance of cooperation in nature, showcasing the intricate web of connections that sustain life across ecosystems. This enduring interplay remains a key factor in the ongoing evolution of diverse species. The intricate relationships formed within this context reveal mutualism as a cornerstone of ecological dynamics. Specific interactions between plants and microbes illustrate this phenomenon vividly. Mycorrhizal fungi, for example, engage in a symbiotic alliance with the roots of various plants, enhancing nutrient uptake, particularly phosphorus, while gaining carbohydrates in return. This exchange fosters not only individual plant health, but also contributes to broader ecosystem stability. In moist environments, interactions between legumes and nitrogen-fixing bacteria further exemplify this collaboration. The bacteria inhabit specialized structures within plant roots, converting atmospheric nitrogen into a usable form. This process enriches the soil, benefiting neighboring flora and promoting biodiversity. The presence of these microbes can lead to healthier ecosystems, as their activities influence soil composition and nutrient cycling. These relationships extend beyond single-species interactions. Complex networks of plants and microbes facilitate nutrient sharing and resilience against pathogens. This interconnectedness underscores the importance of preserving diverse microbial communities, as their functions are crucial for plant health and ecosystem integrity. The dynamics of these mutualistic interactions illuminate the delicate balance of life. By fostering cooperation rather than competition, they demonstrate how cooperation among different life forms can drive evolutionary advancements and sustain ecological systems over time. This understanding reinforces the significance of mutualism in the fabric of life on Earth. The presence of endosymbiotic relationships provides a crucial insight into the emergence of multicellularity. Organisms that once functioned independently began to form symbiotic connections, enabling a new level of complexity. These interactions paved the way for the integration of distinct cellular functions, allowing for specialization within a shared environment. In this context, a fusion of separate entities led to the development of eukaryotic cells, distinguished by their compartmentalized structures. The incorporation of mitochondria and chloroplasts, once free-living prokaryotes, exemplifies this evolutionary leap. These organelles became integral to energy production and photosynthesis, respectively, enhancing the metabolic capacities of their host cells. Over time, cellular cooperation fostered increased efficiency and adaptability, setting the stage for more intricate life forms. The transition from unicellular to multicellular organisms was gradual, marked by incremental changes in cellular communication and coordination. This evolution allowed cells to work together, leading to the formation of tissues and gore. In this intricate web of life, the endosymbiotic theory serves as a foundational principle, illustrating how interdependence can drive innovation and diversity. The legacy of these early partnerships continues to shape the biosphere, influencing the trajectory of life on Earth. This intermingling of distinct life forms has fostered an extraordinary diversity, leading to the emergence of complex eukaryotic organisms. The integration of prokaryotic cells as organelles instigated several evolutionary advancements, including the development of mitochondria and chloroplasts. These organelles have become essential for cellular respiration and photosynthesis, processes that underpin the energy systems of countless species. The resulting energy efficiency allowed organisms to occupy a broader range of ecological niches, enhancing survival and reproduction. This adaptability led to a proliferation of life forms, from simple unicellular entities to intricate multicellular organisms. In the oceans, the symbiotic relationships between corals and photosynthetic algae have created vibrant reefs, serving as vital ecosystems that support immense biodiversity. Terrestrial life has also benefited from these partnerships. Plants, by harnessing solar energy through chloroplasts, have facilitated the evolution of herbivores, which in turn support predators. Such interdependencies illustrate the intricate web of life sustained by these foundational symbiotic events. The endosymbiotic theory underscores a pivotal moment in evolutionary history, emphasizing how cooperation can yield complex systems. This phenomenon not only enhances individual fitness, but also drives the ongoing diversification of life, ensuring that ecosystems remain resilient and adaptable amidst changing environmental conditions. The interconnectedness of life speaks to the enduring legacy of these ancient cellular alliances. The role of endosymbiosis extends beyond simple cellular partnerships, influencing the intricate web of human evolution. Mitochondria, often referred to as the powerhouses of the cell, originated from free-living prokaryotes that entered a symbiotic relationship with ancestral eukaryotic cells. This transition enabled the emergence of complex multicellular organisms, laying the groundwork for subsequent evolutionary advancements. The energy production facilitated by these organelles allowed early eukaryotes to thrive in diverse environments, fostering greater adaptability. These cellular alliances initiated a cascade of evolutionary events. As organisms evolved, the genetic material of the engulfed prokaryotes integrated into the host genome. This genetic exchange resulted in novel pathways and functionalities, enhancing survival prospects. The impact of endosymbiosis is particularly evident in the human lineage, where the incorporation of mitochondria paved the way for the development of larger brains and more sophisticated physiological processes. Beyond energy production, the relationship between host and symbiont has played a crucial role in metabolic diversity. The evolution of human digestion, for instance, showcases how symbiotic bacteria contribute to the breakdown of complex carbohydrates, facilitating nutrient absorption. Such relationships underscore the significance of cooperation in evolution, where mutual benefits drive the development of complex life forms. These complexities extend to the microscopic world, where endosymbiotic bacteria play a crucial role in digestion within the gastrointestinal tracts of many organisms. These bacteria establish a symbiotic relationship that enhances nutrient absorption. This region of biological interaction highlights the intricacies of cohabitation, where host organisms rely on their microbial companions for essential processes. Endosymbiotic bacteria facilitate the breakdown of complex carbohydrates, transforming them into simpler, absorbable sugars. This conversion not only provides energy but also influences the overall health and well-being of the host. In some cases, these microorganisms synthesize vitamins and other nutrients, further enriching the host's diet. The delicate balance within this system is vital. Any disruption can lead to detrimental effects on both the bacteria and the host organism. The mechanisms of these interactions illustrate a finely tuned evolutionary process. Over millions of years, host organisms have adapted to rely on these bacteria for survival, leading to a mutualistic relationship that is both intricate and essential. This symbiosis exemplifies the underlying principles of evolution, where cooperation and adaptation pave the way for greater complexity in life forms. Such interdependencies can be observed across various species, reflecting a broader ecological framework that sustains life on Earth. The intricate relationships within coral reefs exemplify this ecological framework, where diverse organisms coexist and rely on one another for survival. Coral polyps, tiny marine animals, form the foundation of these vibrant ecosystems. They harbor symbiotic algae known as zooxanthellae within their tissues. This partnership allows the polyps to obtain energy through photosynthesis, a process that significantly contributes to the reef's growth and resilience. These reefs serve as a crucial habitat for myriad marine species, providing shelter and nourishment. The presence of zooxanthellae not only aids coral nutrition, but also enhances the biodiversity of the surrounding environment. Fish, mollusks, and crustaceans thrive in these complex structures, establishing a network of interdependence that maintains the delicate balance of the ecosystem. And coral reefs play a vital role in coastal protection, acting as natural barriers against storm surges and erosion. Their physical structure dissipates wave energy, helping to shield shorelines from the forces of nature. The health of these reefs is indicative of broader oceanic conditions, as they are sensitive to changes in temperature, acidity, and pollution. The decline of coral reefs due to climate change and human activity threatens not only the myriad life forms they support, but also the ecological services they provide to coastal communities. Understanding these relationships emphasizes the importance of preserving such ecosystems for future generations. The impact of endosymbiosis on animal physiology extends into the very foundation of metabolic processes. This merging of distinct cellular entities created complex energy pathways that transformed the way organisms harness and utilize energy. Mitochondria, once free-living bacteria, evolved into essential organelles within eukaryotic cells, facilitating aerobic respiration. This adaptation allowed for increased energy production, which is crucial for the growth and survival of multicellular organisms. Chloroplasts, derived from photosynthetic bacteria, similarly contributed to the ability of certain organisms to convert sunlight into chemical energy. This process not only supports individual life forms, but also underpins entire ecosystems by providing energy for plants, which in turn sustain herbivores and a myriad of other species. It is this evolutionary leap that enabled greater specialization among cells, leading to the diverse physiological characteristics observed across the animal kingdom. Organisms developed intricate systems for nutrient uptake, waste elimination, and cellular communication, all of which are influenced by their symbiotic origins. The presence of these organelles facilitates a higher degree of metabolic efficiency, allowing for the complexity seen in higher life forms. With the intricate interplay of these cellular components, the foundation of animal physiology is fundamentally rooted in the ancient partnerships formed through endosymbiosis, highlighting a remarkable chapter in the evolutionary narrative. The foundation of this symbiotic partnership extends beyond animal physiology, influencing the intricate relationships observed in fungi and plants. Fungi often establish mutualistic associations with plant roots, forming mycorrhizal networks that enhance nutrient exchange and facilitate water absorption. These networks serve as conduits, allowing the transfer of vital resources between different plant species, fostering collaboration in nutrient-poor soils. In this region, the fungi receive carbohydrates produced through photosynthesis, while plants benefit from increased access to essential minerals and improved resistance to pathogens. The endosymbiotic origins of these interactions underscore the evolutionary significance of cooperation in sustaining diverse ecosystems. Examining lichens reveals another dimension of this symbiosis, comprising fungi and photosynthetic partners such as algae or cyanobacteria. Lichens thrive in some of the harshest environments. The fungal component provides structure and protection, while the photosynthetic partner generates organic compounds through photosynthesis. This cooperative arrangement enables lichens to colonize rocky surfaces and withstand extreme temperatures, illustrating the adaptability of these organisms through collaborative evolution. The role of endosymbiosis extends to the plant kingdom as well, influencing the evolution of cellular structures such as chloroplasts. These organelles, derived from ancient cyanobacterial endosymbionts, are essential for photosynthesis, powering the energy demands of plant life. Thus, these partnerships illustrate the profound impact of endosymbiotic relationships across various forms of life. Artificial selection provides a controlled environment to explore endosymbiotic relationships. Through this method, researchers manipulate genetic traits within organisms, fostering symbiotic interactions that might not occur in nature. By selecting specific characteristics, it becomes possible to enhance the efficiency of photosynthesis in plant cells. For example, this modification can replicate the evolutionary advantages seen in natural endosymbionts. Observations reveal that when certain algae are introduced to various host organisms, the resulting interactions may lead to increased energy production. These experiments create a unique opportunity to study how selected traits influence the stability and functionality of symbiotic partnerships. The outcome often results in enhanced resilience to environmental stresses, illuminating the potential benefits of such relationships. In laboratory settings, the introduction of bacteria into protozoa enables scientists to observe the dynamics of cooperation at a cellular level. These interactions often result in mutual benefits, showcasing how one organism can provide essential resources while the other offers protection or additional metabolic capabilities. Researchers meticulously document these changes, measuring growth rates, reproductive success, and the overall health of the symbiotic pairings. As understanding deepens, it becomes evident that the principles underlying endosymbiosis extend beyond natural history. Each experiment offers insight into the complex web of life, revealing how cooperation can drive evolutionary processes across diverse ecosystems. The intricate relationship between endosymbiosis and disease manifests in unexpected ways. Pathogens often exploit cellular machinery, integrating their own genetic material into host systems. This biological interplay can lead to the emergence of new viral strains or antibiotic-resistant bacteria. Understanding these mechanisms enhances comprehension of disease dynamics, illustrating how symbiotic relationships can shift towards pathogenicity. In this context, endosymbiotic organisms may evolve alongside their hosts, creating a delicate balance. When environmental stresses disrupt this balance, the once beneficial relationship may transform, leading to opportunistic infections. The reprogramming of cellular functions by symbiotic entities can result in altered immune responses, facilitating the proliferation of harmful agents within a host. Research indicates that certain symbionts can modulate host physiology, influencing susceptibility to pathogens. This phenomenon is particularly evident in various insect species, where microbial communities contribute to immunological defenses. The loss or alteration of these symbionts can leave hosts vulnerable, triggering cascading effects on population dynamics. On a larger scale, studying these interactions provides insight into evolutionary trends. The co-evolution of symbionts and pathogens exemplifies the adaptability of life, showcasing how cellular partnerships can reshape ecosystems. Such insights pave the way for new therapeutic strategies, emphasizing the significance of understanding endosymbiotic relationships in combating diseases that threaten global health. The exploration of endosymbiotic theory extends far beyond the confines of evolutionary biology, impacting various facets of biotechnology. This perspective invites scientists to harness the power of symbiotic relationships to develop innovative biotechnological applications. By understanding how eukaryotic cells evolved through the incorporation of prokaryotic organisms, researchers can engineer synthetic organisms that mimic these natural partnerships. This approach fosters the creation of biofactories capable of producing biofuels, pharmaceuticals, and other valuable materials. In this context, genetically modified organisms can be designed to perform specific functions, such as enhancing nutrient absorption in agricultural crops, or improving the efficiency of waste degradation in environmental applications. These engineered systems could lead to sustainable practices that minimize the need for chemical fertilizers or pesticides, promoting ecological balance. The implications stretch into the realms of medicine as well. By studying the endosymbiotic relationships that underpin cellular functionality, biotechnologists can develop targeted therapies that leverage these interactions to improve drug delivery systems. This understanding allows for the design of treatments that are more efficient and tailored to individual patient needs. Such advancements not only have the potential to revolutionize various industries, but also encourage a deeper understanding of life's intricate connections. These insights remind humanity of nature's wisdom, urging careful consideration of how to integrate these discoveries into future innovations. The role of endosymbiosis significantly shaped plant evolution and adaptation. The incorporation of photosynthetic prokaryotes into ancestral eukaryotic cells marked a pivotal moment in the history of life. This process gave rise to chloroplasts, organelles that enable plants to harness sunlight and convert it into energy. This fundamental transformation allowed for the emergence of diverse plant life across various ecosystems. In this region of evolution, early photosynthetic organisms thrived in environments where light was abundant, gradually spreading their genetic material through generations. Over time, these adaptations led to the development of structures such as leaves and stems, optimized for light capture and nutrient transport. The relationship between host cells and their endosymbionts exemplifies a profound interdependence, illustrating how cooperation can drive complexity in biological systems. Variations in environmental conditions prompted a multitude of evolutionary pathways in aquatic habitats. Some plants adapted to life underwater, developing specialized features such as floating leaves and gas-filled bladders. Terrestrial adaptations led to the formation of protective cuticles and advanced reproductive strategies. This remarkable flexibility allowed plants to colonize diverse habitats, from arid deserts to lush rainforests, showcasing their resilience and innovative survival strategies. Each evolutionary step reflected an intricate balance between internal capabilities and external pressures, underscoring the profound impact of endosymbiosis on the plant kingdom. Shifts in climate impose considerable stress on ecosystems, influencing the delicate relationships formed through endosymbiosis. Alterations in temperature and precipitation patterns affect the metabolic rates of symbiotic partners, leading to changes in nutrient exchange and energy flow. This region experiences variability, causing disruptions in the mutualistic interactions that have evolved over millions of years. Extremes of heat can inhibit the performance of photosynthetic organisms, impacting their ability to sustain the systems that rely on them. Vulnerable partners may struggle to adapt, resulting in a decline of biodiversity and altering community structures. These modifications ripple through the ecosystem, where every organism plays a role in maintaining balance. Observations indicate that some symbiotic relationships exhibit resilience, adapting to new environmental conditions, while others falter under pressure. The oceanic realm serves as a critical example, where coral reefs provide habitats for diverse marine species. Rising sea temperatures lead to coral bleaching, disrupting the symbiotic relationship with zooxanthellae. This loss jeopardizes not only the coral, but also the myriad of life forms dependent on this habitat. The interdependence inherent in these relationships highlights the fragility of ecological systems, revealing how climate change can unravel centuries of evolutionary innovation. Emphasizing the need for a deeper understanding of these dynamics in the face of global shifts. This relationship fosters an intriguing perspective on astrobiology. The endosymbiotic theory suggests that complex life forms may not only arise from random mutations, but also through cooperative interactions among simpler organisms. Such cooperative behaviors could provide the framework for life beyond Earth, where conditions may favor similar symbiotic partnerships. Investigations into extreme environments on our planet, such as deep-sea hydrothermal vents and acidic hot springs, offer insight into how life can thrive under adverse conditions. These ecosystems, reminiscent of potential extraterrestrial environments, hint at the possibility of life forms that rely on mutualistic relationships. Such life forms could represent a fundamental strategy for survival in the universe. The presence of organelles like mitochondria and chloroplasts, which originated from independent prokaryotic organisms, underscores the potential for evolutionary innovation through endosymbiosis. This phenomenon may not be limited to Earth. Rather, it may be a universal mechanism that facilitates the emergence of complex life elsewhere in the cosmos. Astrobiological research is increasingly focused on identifying exoplanets within habitable zones where conditions could allow the establishment of such symbiotic relationships. The study of life's adaptability and resilience on Earth informs predictions about its potential forms and functions in environments yet unexplored, expanding the horizons of scientific inquiry into the vast unknown. Endosymbiotic relationships have not only shaped the origins of complex life, but have also played a pivotal role in the evolution of the immune system. This phenomenon allowed certain cells to incorporate others, creating a new level of biological complexity. The merger of prokaryotic and eukaryotic cells led to the development of organelles, which provided additional functions and enhanced metabolic capabilities. Consequently, this evolutionary leap enabled the emergence of sophisticated immune responses. The innate immune system, characterized by its immediate and non-specific reactions, reflects the consequences of this cellular collaboration. It employs a range of mechanisms, such as phagocytosis, to identify and eliminate pathogens. The continued evolution of this system demonstrates how endosymbiotic events facilitated greater adaptability to diverse microbial challenges as these cells evolved. they began to utilize signaling pathways that were once solely associated with their endosymbiotic partners. This intricate interplay between cells has given rise to the adaptive immune system, which provides a more targeted response to specific pathogens. The formation of lymphocytes, crucial for immunological memory, can also be traced back to these symbiotic relationships, underscoring their lasting impact on the immune landscape. Exploring the evolutionary lineage reveals a profound connection between endosymbiosis and the dynamic nature of immune responses, emphasizing the intricate web of interactions that underpin life on Earth. Mitochondrial genetics serves as a compelling example of endosymbiosis's implications. The presence of distinct mitochondrial DNA, separate from nuclear DNA, highlights a remnant of ancestral prokaryotic organisms that once merged with primitive eukaryotic cells. This genetic material, generally circular and lacking the complex packaging of chromosomal DNA, reflects its origins and offers insights into evolutionary history. Each mitochondrion, functioning as the powerhouse of the cell, contributes to energy production through oxidative phosphorylation. This process is not merely a biochemical reaction. It is a testament to the integration of once independent organisms. Genetic variations within mitochondrial DNA can yield significant differences in metabolic efficiency and adaptability, influencing overall cellular health and functioning. The maternal inheritance of mitochondrial DNA underscores its role in evolutionary studies, tracking lineage and migration patterns across generations. Such genetic continuity presents a unique perspective on human evolution, revealing connections that span continents and millennia. Research into mitochondrial genetics also extends to understanding diseases, as mutations can lead to a variety of conditions impacting energy metabolism and cellular respiration. This link between genetic heritage and contemporary health outcomes emphasizes the continuing relevance of endosymbiotic events, further illustrating the intricate interdependence that characterizes life. These insights reflect the enduring legacy of ancient cellular alliances. The role of endosymbiosis extends beyond mere cellular evolution, intertwining with the intricate networks of biogeochemical cycles in various ecosystems. These symbiotic relationships facilitate essential processes such as carbon fixation and nutrient cycling. Photosynthetic eukaryotes, derived from ancestral symbiotic events, contribute significantly to the absorption of atmospheric carbon dioxide. This conversion into organic matter serves as a foundation for food webs, supporting an array of life forms. In aquatic environments, phytoplankton, descendants of endosymbiotic organisms, play a pivotal role in global carbon cycles. These microscopic entities harness sunlight, transforming inorganic carbon into biomass. The subsequent sinking of organic material to the ocean floor aids in carbon sequestration, effectively removing carbon dioxide from the atmosphere over long timescales. Nitrogen-fixing bacteria, often forming symbiotic relationships with plant roots, also demonstrate the importance of these ancient alliances. Through the process of nitrogen fixation, they convert atmospheric nitrogen into forms usable by plants, enhancing soil fertility and promoting diverse ecosystems. The impact of endosymbiosis reaches into the soil as well, where the interactions between fungi and plant roots, known as mycorrhizal associations, lead to improved nutrient uptake and soil structure. Such relationships underscore the interconnectedness of life and the biochemical processes that sustain it. Advances in molecular techniques provide unprecedented insights into endosymbiosis. Techniques such as next-generation sequencing allow researchers to unravel the complex genomes of both host and symbionts. This capability facilitates the identification of genes that contribute to the symbiotic relationship, revealing how these organisms have adapted over evolutionary timescales. High-throughput gene expression analysis further highlights the dynamic interactions between cells, showcasing how they communicate and respond to environmental stimuli. Optogenetics stands at the forefront of experimental methodologies, enabling scientists to manipulate cellular functions with light. It offers a way to observe real-time interactions within endosymbiotic systems, uncovering the nuances of cellular behavior and the biochemical pathways involved. Such precision aids in understanding how these organisms cooperate, share resources, and mechanisms, the study of small molecule metabolites, adds another layer of complexity. By profiling metabolic changes in both partners, it becomes possible to trace the flow of nutrients and energy within the symbiotic relationship. This insight reveals the metabolic integration that characterizes successful endosymbiosis. Microscopy techniques, particularly advanced imaging methods, allow for the visualization of these intricate interactions at cellular and subcellular levels. These observations illuminate the physical relationships formed during endosymbiosis, providing a window into the evolutionary history that defined the emergence of eukaryotic life. This understanding reshapes conservation strategies by highlighting the intricate connections among diverse life forms, recognizing that eukaryotic cells originated through symbiotic relationships encourages a holistic approach to ecosystem preservation. Each organism plays a pivotal role within its environment, much like the cellular partners that led to complex life. Efforts to conserve biodiversity benefit from this theory, emphasizing the necessity of protecting not just individual species but entire ecosystems. Disruption in one area can reverberate through interconnected systems, fundamentally altering the balance of life. This perspective fosters a greater appreciation for even the smallest organisms, which may hold the key to sustaining larger ecological networks. The theory also informs genetic research, offering insights into how symbiotic relationships can enhance resilience against environmental stresses. By understanding the molecular basis of these interactions, scientists can develop strategies to bolster ecosystems in the face of climate change. Such insights lead to innovative conservation practices, including the restoration of habitats that support symbiotic relationships, ensuring that these interactions continue to thrive. The implications extend beyond mere survival. They highlight the potential for evolutionary innovation within conservation programs. Monitoring these dynamics allows for adaptive management strategies, ensuring that interventions remain effective as ecosystems evolve in response to shifting conditions. This nuanced understanding of life's origins. The role of endosymbiotic relationships significantly enhances agricultural practices, illustrating the interconnectedness of various organisms. Beneficial interactions between plants and their microbial partners optimize nutrient uptake and improve resilience against environmental stressors. Specific bacteria and fungi form associations with plant roots, facilitating the exchange of essential nutrients like nitrogen and phosphorus. This symbiosis, certain crops, such as legumes, actively engage in endosymbiosis with nitrogen-fixing bacteria. This process not only enriches the soil, but also reduces the need for synthetic fertilizers, contributing to sustainable farming practices. The presence of mycorrhizal fungi further exemplifies beneficial relationships, enhancing water absorption and nutrient acquisition for many plant species. These fungi form extensive networks in the soil, connecting various plants, allowing them to share resources and communicate stress signals. In addition to enhancing nutrient availability, endosymbiotic relationships also contribute to pest resistance. Some plants produce chemical signals that attract beneficial insects when under threat, creating a natural defense mechanism. This integrated approach to agriculture reflects a deeper understanding of ecological dynamics, emphasizing the importance of maintaining biodiversity and fostering these relationships. Through the lens of endosymbiotic theory, agricultural methods can evolve, promoting sustainability and resilience in food systems, while reducing the reliance on chemical inputs. This ongoing exploration continues to reveal the intricate web of life that sustains both plants and animals. Future research on endosymbiosis aims to deepen understanding of the evolutionary processes that led to the integration of distinct cellular organisms. Investigations are shifting towards the genomic analysis of symbiotic relationships, with an emphasis on identifying specific genes that facilitate cooperation between host cells and their endosymbionts. The implications of such studies extend beyond mere cellular function. They may illuminate the adaptive advantages conferred by endosymbiotic arrangements, potentially revealing how such relationships influenced the trajectory of complex life. Recent advancements in sequencing technology provide unprecedented insights into the genetic material of these organisms. These tools enable scientists to explore the phylogenetic relationships among various species, tracing back the lineage of endosymbiotic events that shaped the diversity of life. The study of protists and their endosymbionts has gained particular interest, revealing how these interactions can lead to unique metabolic capabilities. Laboratory experiments are increasingly focusing on synthetic biology, attempting to recreate endosymbiotic relationships to observe the effects on cellular behavior and function. This approach could yield significant breakthroughs in biotechnology, including advancements in biofuels and sustainable agriculture. The intersection of endosymbiotic theory and genetic engineering presents an intriguing frontier in biological science. Genetic engineering has the potential to manipulate the genomes of organisms, facilitating the exploration of cellular evolution and symbiotic relationships. By introducing specific genes into host cells, researchers can observe the impact on cellular functions, revealing insights into how these relationships may have developed over millennia. Investigations into organelle origins provide a deeper understanding of how chloroplasts and mitochondria function as integral components of eukaryotic cells. These organelles possess their own genetic material, which shares similarities with that of prokaryotes. This observation underscores the significance of horizontal gene transfer, a phenomenon that may have played a vital role during the early stages of cellular evolution. The ability to engineer genetic pathways could enhance the efficiency of energy production and metabolic processes within these organelles. By optimizing these functions, it may be possible to develop organisms capable of greater resilience in fluctuating environmental conditions. This research could lead to organisms that not only thrive in their ecological niches, but also contribute to a balanced ecosystem through enhanced nutrient cycling and energy flow. The implications extend beyond theoretical frameworks. Practical applications in agriculture and renewable energy could emerge, demonstrating the relevance of evolutionary theory in addressing contemporary challenges. Phylogenetic advances have significantly enhanced the exploration of endosymbiotic relationships, facilitating a deeper understanding of these complex interactions. High-throughput sequencing techniques enable scientists to decode genomic data at an unprecedented scale, revealing the intricate genetic exchanges between host cells and their symbiotic partners. These methodologies have illuminated the evolutionary pathways that led to the formation of organelles, such as mitochondria and chloroplasts, showcasing their origins from free-living bacteria. In this context, bioinformatics tools play a crucial role in managing and analyzing the vast amounts of genomic data produced by employing machine learning algorithms. researchers can identify patterns and predict interactions within microbial communities, providing insights into how these relationships evolve over time. This technological progress is not only advancing fundamental biology but also paving the way for biotechnological innovations. The development of synthetic biology techniques allows for the manipulation of microbial genomes, enabling scientists to engineer symbiotic relationships that could optimize productivity in agriculture and bioenergy. These engineered systems can enhance nutrient absorption or increase stress resistance in crops, demonstrating the practical potential of endosymbiotic principles. In laboratory settings, advanced microscopy techniques visualize these interactions at the cellular level, revealing the dynamic nature of symbiosis. This combination of tools fosters a comprehensive understanding of how symbiotic relationships shape ecosystems and influence evolutionary trajectories. The foundation of evolutionary developmental biology rests upon the principles of endosymbiosis, illustrating how distinct cellular entities can merge to create more complex forms of life. This process has far-reaching implications, particularly in the development of multicellular organisms. By integrating specialized functions from one another, these once independent cells enabled the emergence of intricate cellular structures and functions. The incorporation of mitochondria and chloroplasts into eukaryotic cells is a prime example. These organelles, originally free-living bacteria, provide essential energy production capabilities. Such energy efficiency supports the greater complexity seen in eukaryotes, allowing for diverse forms of life, from simple algae to complex mammals. This evolution of cellular architecture is vital, as it has led to the differentiation of tissue types and specialized organs. Research continues to unveil the genetic exchanges that occur during endosymbiotic events, revealing a dynamic relationship between the host and the symbionts. This interplay not only enhances survival but also drives innovation within biological systems. The evolutionary potential witnessed in various lineages showcases the importance of cooperation over competition, emphasizing a paradigm shift in understanding life's development. Investigating these relationships provides insight into how organisms adapt to their environments. It reflects an ongoing journey of evolution marked by collaboration, adaptation, and profound biological transformation. This journey invites contemplation within the philosophy of science, exploring the implications of shared existence and mutual benefit in evolutionary processes. The endosymbiotic theory posits that complex cells, known as eukaryotes, emerged through the integration of simpler prokaryotic organisms. This notion challenges traditional views of evolution that often emphasize competition and survival of the fittest. It highlights a paradigm shift, suggesting that cooperation can be a powerful force of change. Ancient microorganisms, once independent, adapted to new roles within host cells. The theory's development illustrates the dynamic relationship between observation and interpretation. It required the synthesis of evidence from various fields, including genetics, biochemistry, and paleontology, leading to its eventual recognition. Such interdisciplinary connections reflect the collaborative essence of scientific inquiry, revealing that knowledge is often built upon the foundations laid by diverse perspectives. In contemplating the endosymbiotic theory, one recognizes the importance of adaptability in both organisms and ideas. Evolutionary biology thus serves not only as a study of living systems, but also as a mirror, reflecting the complexities and interconnections inherent in the pursuit of understanding the universe. Cellular communication emerges as a critical component in the narrative of endosymbiosis. The intricate exchanges between cells reflect a profound evolution of life forms, laying the groundwork for more complex organisms. This phenomenon showcases how two distinct entities can merge to create a unified system, enhancing their adaptive capabilities. The process exemplifies the transition from solitary existence to cooperative interaction, paving the way for multicellularity. In this context, signaling mechanisms become essential. Various molecules facilitate communication, enabling cells to coordinate activities crucial for survival. These signals, whether chemical or electrical, allow for the sharing of resources and information, thus fostering a sense of community within the cellular environment. The evolution of these mechanisms can be traced back to early symbiotic relationships, where one organism provided energy or protection, while the other contributed mobility or nutrient acquisition. This region of study reveals the layers of complexity involved in the development of cellular networks. Analyzing these interactions offers insights into the early dynamics of life on Earth. The transformation of independent cells into interdependent units signifies a pivotal shift in evolutionary history, highlighting the importance of cooperation over competition. As the understanding deepens, it becomes evident that these early symbiotic events were foundational in shaping not only cellular architecture, but also the very fabric of biological evolution. Evolutionary rates within the context of endosymbiosis exhibit intriguing patterns that reflect the complexities of these ancient partnerships. The integration of prokaryotic cells into eukaryotic hosts catalyzed significant physiological changes, influencing not only metabolic processes, but also genomic evolution. This relationship suggests a dynamic interplay between cooperation and competition, where evolutionary pressures could accelerate adaptation. In this intricate environment, the genetic material of the engulfed organisms often became integrated into the host genome, leading to innovations in cellular processes. Such events can be traced through phylogenetic studies, revealing instances where evolutionary rates surged following the establishment of symbiotic relationships. The transfer of genes conferred distinct advantages, allowing these newly formed cells to exploit diverse ecological niches effectively. Observations in various lineages indicate that organisms resulting from endosymbiotic events tend to exhibit diversified traits at a faster rate compared to their non-symbiotic counterparts. This phenomenon is particularly evident in plant and animal lineages, where chloroplasts and mitochondria have played critical roles in energy production and photosynthesis, respectively. The unique genetic contributions from these endosymbionts have thus sculpted evolutionary trajectories, allowing for enhanced adaptability in fluctuating environments. Such insights into the relationship between endosymbiosis and evolutionary rates deepen the understanding of life's complexity on Earth. Each event contributes to the rich history of biological diversity. The interplay between cellular interactions and evolutionary processes illustrates the significance of the endosymbiotic theory in shaping modern evolutionary synthesis. This theory posits that certain organelles within eukaryotic cells, such as mitochondria and chloroplasts, originated from free-living prokaryotic organisms that entered into a symbiotic relationship with ancestral eukaryotes. This integration fundamentally altered cellular function and energy production, enabling the emergence of complex life forms. Evidence supporting this theory is found in the genetic similarities between these organelles and their prokaryotic ancestors, suggesting a shared evolutionary lineage. The distinct circular DNA structures within mitochondria and chloroplasts mirror those of bacteria, reinforcing the notion of their endosymbiotic origins. This relationship not only exemplifies cooperation and mutualism, but also showcases the adaptability of life forms over extensive periods. The implications of the endosymbiotic theory extend into various domains of biology, influencing our understanding of genetic exchange and evolutionary innovation. The merging of distinct cellular entities highlights a pivotal moment in evolutionary history, where the capacity for diversification increased significantly. This event laid the groundwork for the vast array of multicellular organisms that inhabit Earth today. revealing the interconnected nature of life. The framework of modern evolutionary synthesis continues to be enriched by these insights, shaping ongoing research and understanding of life's complexity and resilience. The intricate relationship between humans and their microbial inhabitants plays a crucial role in overall health. These microorganisms, residing predominantly in the gut, contribute significantly to digestion, metabolism, and immune function. This region of the human body serves as a bustling ecosystem where trillions of bacteria, fungi, and viruses coexist, interacting with one another and with their host. Research indicates that the composition of this microbial community can influence various aspects of health, including susceptibility to diseases and the efficacy of treatments. Diverse microbial populations are associated with balanced bodily functions, while dysbiosis or imbalance can lead to conditions such as obesity, diabetes, and inflammatory bowel disease. The interplay between dietary habits and microbial health highlights the importance of nutrition in maintaining this delicate equilibrium. Recent studies further explore the mechanisms by which these microorganisms communicate with human cells. They produce metabolites that can affect mood, cognition, and even immune responses. Understanding these interactions is essential for developing innovative therapeutic approaches, including probiotics and personalized dietary interventions, aimed at restoring microbial balance. The ongoing research in this field continues to unveil the complexity of these relationships, emphasizing the integral role that microbes play in shaping human health. This perspective enriches our understanding of health and disease, offering pathways toward holistic approaches in medicine. The emergence of extremophiles illustrates the profound impact of endosymbiosis on evolutionary pathways. These remarkable organisms thrive in environments previously deemed inhospitable, such as hydrothermal vents, acidic hot springs, and hypersaline lakes. Their existence reveals the adaptive potential of life when cellular cooperation takes precedence over competition. Through endosymbiotic relationships, primitive cells gained new metabolic capabilities, enabling them to harness energy from extreme surroundings. In this context, the incorporation of prokaryotic cells into ancestral eukaryotes marked a turning point. The resulting cellular structures facilitated novel biochemical processes, allowing these organisms to exploit unique ecological niches. The ability to endure extreme temperatures or salinity levels can be traced back to these symbiotic integrations, which enhanced resilience and adaptability. Research indicates that the genetic material of endosymbionts has been retained in many extremophiles, contributing to their evolutionary success. The shared genetic heritage fosters a diverse array of metabolic pathways, granting these organisms the ability to thrive under harsh conditions. This evolutionary strategy underscores the significance of cooperation among cells, shaping not only individual species but entire ecosystems. As scientists continue to explore the complex relationships born from endosymbiosis, the implications for understanding life's resilience in extreme conditions grow ever clearer. The study of extremophiles stands as a testament to the power of symbiotic evolution in shaping the tree of life. This evolutionary process provides insight into how organisms adapt to diverse environments. Endosymbiosis serves as a mechanism through which cells can acquire new functionalities, enabling them to thrive under conditions that may otherwise be inhospitable. The incorporation of prokaryotic cells into ancestral eukaryotic organisms illustrates this principle. By forming a mutualistic relationship, these once independent entities transformed their host, granting it enhanced metabolic capabilities. In aquatic ecosystems, for instance, The symbiotic relationship between marine invertebrates and photosynthetic algae allows these organisms to harness sunlight, converting it into energy. This adaptation not only supports their survival, but also contributes to the overall productivity of their habitat. Likewise, the association between certain fungi and plant roots facilitates nutrient absorption, enabling plants to flourish in nutrient-poor soils. These interactions exemplify the profound impact of endosymbiosis on ecological dynamics. The resulting organisms possess traits that allow them to exploit resources more efficiently and adapt to changing environmental conditions. In extreme environments, such as hydrothermal vents or acidic lakes, the ability to integrate different metabolic pathways can mean the difference between survival and extinction. The legacy of this cellular collaboration echoes through evolutionary history, shaping the complex web of life that persists today. This fundamental process continues to offer insights into the resilience of life on Earth. The exploration of synthetic biology opens new avenues for understanding the principles of endosymbiosis. Researchers are investigating the potential of engineering cells to mimic the intricate relationships observed in nature. This region of study seeks to create organisms that can perform specific tasks, enhancing agricultural productivity and addressing environmental challenges. The manipulation of genetic material allows for the incorporation of traits found in different species, reminiscent of the merging that defines endosymbiotic relationships. The implications of such advancements extend beyond mere biotechnology. They may lead to the development of synthetic cells capable of energy production, carbon capture, or even bioremediation. These innovations draw inspiration from the ancient processes that shaped modern eukaryotic life. By understanding how two distinct entities can coalesce into a singular, cohesive system, scientists are paving the way for novel applications that could transform ecosystems. Laboratories are beginning to replicate the conditions that fostered early endosymbiotic events, which may lead to breakthroughs in cellular efficiency and adaptability. The prospect of creating life forms that not only survive but thrive in various environments represents a significant leap in biological understanding. This realm of synthetic biology, entwined with endosymbiotic theory, holds the promise of reshaping future biological landscapes, fostering sustainability and resilience in the face of global challenges. The interconnectedness of life is a fundamental principle illustrated by endosymbiotic theory. This concept reveals how disparate organisms can combine, creating complex cellular structures through cooperation rather than competition. Mitochondria and chloroplasts, once free-living prokaryotes, became integral components of eukaryotic cells, exemplifying how symbiosis can drive evolutionary innovation. This evolutionary event marks a pivotal moment when two entities merge to enhance energy production and photosynthesis, leading to the rich diversity of life as we know it today. These relationships emphasize that life is not an isolated phenomenon, but rather a continuum of interactions. Ecosystems thrive on these connections, where organisms rely on one another for sustenance and survival. The delicate balance maintained in these systems underscores the importance of each participant, highlighting that the health of one is often intertwined with the health of all. Lessons from this theory extend beyond biological frameworks, informing ecological conservation efforts and sustainable practices. Understanding that cooperation can yield resilience offers pathways to address environmental degradation. This perspective invites a re-evaluation of human approaches to nature, suggesting that fostering symbiotic relationships can lead to more sustainable living. In essence, the legacy of endosymbiosis teaches that unity in diversity can pave the way for adaptation and survival in an ever-changing world. The relationship established through endosymbiosis extends beyond mere survival, influencing the emergence of antibiotic resistance within microbial communities. This process, wherein one organism resides within another, provides a unique environment for genetic exchange and adaptation. In this context, bacteria often acquire resistance genes from their symbiotic partners, enhancing their ability to withstand antibiotic pressures. Survival mechanisms develop within these shared habitats, where the exchange of genetic material becomes a vital strategy. Horizontal gene transfer allows resistant traits to proliferate rapidly, creating populations that can thrive even in the presence of antimicrobial agents. Within these environments, the dynamics of cooperation and competition shape the evolution of resistance, leading to the emergence of resilient strains. These interactions illustrate the interconnectedness of life forms, emphasizing that resistance is not solely a response to external threats, but rather a product of prolonged coexistence. The evolutionary pathways formed through endosymbiotic relationships reveal the complexity of resistance mechanisms, showcasing how diverse organisms adapt in concert to environmental challenges. In this intricate web of interactions, the implications of endosymbiosis extend beyond individual species, influencing broader ecological systems. The adaptations facilitated by these relationships mark a critical aspect of microbial evolution, reshaping our understanding of resistance in the face of modern antibiotic challenges. The intricate relationships fostered through endosymbiosis extend far beyond individual microbial evolution, profoundly influencing marine ecosystems. In this region, symbiotic interactions between organisms such as corals and their photosynthetic partners, dinoflagellates, exemplify this phenomenon. These partnerships enable corals to harness sunlight, converting it into energy, which sustains not only their growth, but also the diverse marine life, relying on coral reefs as vital habitats. As these organisms thrive, they create complex structures that provide shelter and breeding grounds for countless species. The interconnectedness of these systems underlines the significance of endosymbiotic relationships in maintaining biodiversity. The balance of nutrient cycling, facilitated by symbiotic bacteria and algae, fosters a rich environment where various trophic levels can coexist and flourish. In deeper waters, similar symbiotic dynamics can be observed. The reliance of certain marine organisms on chemosynthetic bacteria illustrates how endosymbiosis adapts to varying conditions, such as the absence of sunlight. These relationships enable organisms to survive in extreme environments, showcasing the remarkable versatility of life. The shifting health of these marine ecosystems often reflects changes in the symbiotic relationships within them. Disruptions caused by environmental stresses can lead to the breakdown of these partnerships, highlighting the delicate balance that sustains marine biodiversity. Understanding this influence remains crucial for conservation efforts and the preservation of oceanic life. The emergence of complex traits can be traced back to the fundamental processes of endosymbiosis. This phenomenon, where one organism resides within another, has played a pivotal role in the evolution of eukaryotic cells. These cells, characterized by their membrane-bound organelles, gain significant advantages through the incorporation of symbiotic prokaryotes. This region of cellular evolution illustrates how distinct entities can merge, leading to enhanced metabolic capabilities and resilience. Chloroplasts and mitochondria, the primary examples of such endosymbiotic relationships, provide energy through photosynthesis and respiration. respective. They have not only contributed to the energy dynamics within cells, but have also facilitated the development of complex biochemical pathways. The integration of these organelles allowed for the diversification of life forms, enabling organisms to adapt to various environmental challenges. Through generations, the genetic material of these symbionts has been integrated into the host's genome. This genetic exchange has propelled evolutionary processes, leading to increased complexity in physiological and morphological traits. As these traits evolved, so too did the organism's ecological roles, influencing community structures across ecosystems. On examining the legacy of endosymbiosis, it becomes evident that these relationships were instrumental in shaping the diverse forms of life present today. The intricate interplay of cooperation and adaptation highlights the profound impact of these evolutionary events on the tapestry of biological existence. Cultural perspectives on endosymbiotic relationships reveal a rich tapestry of interpretations and beliefs across various societies. In many ancient cultures, the concept of unity through collaboration resonates with the creation myths that emphasize interconnectedness. For instance, some indigenous traditions recognize the importance of symbiosis in the natural world, likening it to the harmonious relationships within their communities. This analogy reflects an understanding that strength arises from collaboration, mirroring the cellular interactions that underpin life. Modern interpretations often draw parallels between cellular cooperation and human relationships. The idea that individuals and communities thrive through mutual support can be seen in numerous social philosophies. These insights prompt reflections on sustainability and ecological responsibility, urging a deeper appreciation of the relationships that sustain ecosystems. Scientific advancements have further influenced cultural narratives. The endosymbiotic theory, once a groundbreaking hypothesis, has become a foundational concept in biology, prompting discussions surrounding genetic heritage and evolutionary history. As knowledge expands, so does the recognition of the complexity inherent in these cellular alliances. Artistic expressions also embrace themes of symbiosis. Literature, visual arts, and performances convey the notion of interconnectedness, drawing on biological principles to explore existential themes. This exploration fosters a dialogue that transcends disciplinary boundaries, illustrating the enduring relevance of these relationships in shaping both life and culture. The endosymbiotic theory offers a profound insight into the origins of complex life. This principle posits that certain organelles within eukaryotic cells, such as mitochondria and chloroplasts, originated as independent prokaryotic organisms. Over time, a symbiotic relationship developed, leading to a new level of cellular organization. The implications of this theory extend beyond biology, influencing the fields of genetics, evolution, and ecology. In educational contexts, it serves as a vital framework for understanding the interconnectedness of life forms. This perspective encourages students to consider evolutionary processes not as isolated events, but as a continuum of interactions and adaptations. By examining the cellular symbiosis, learners gain insights into how life can emerge from cooperation, rather than solely competition. This concept also highlights the importance of scientific inquiry and critical thinking. As students explore the evidence supporting this theory, they engage with various research methodologies, from molecular analysis to fossil records. Such investigations cultivate a deeper appreciation for the complexity of biological systems. Incorporating the endosymbiotic theory into curricula enriches discussions on biodiversity and the evolutionary tree of life. It illustrates how scientific understanding evolves, shaped by new discoveries. The ongoing exploration of these cellular relationships continues to inspire a sense of wonder regarding life's intricate tapestry. Case studies of endosymbiosis in extremophilic organisms reveal the remarkable adaptability of life. In the depths of hydrothermal vents, certain microorganisms thrive in extreme temperatures and pressures, forming symbiotic relationships that illustrate the principles of endosymbiosis. These organisms have developed mechanisms to harness energy from sulfur compounds, demonstrating the evolutionary benefits of cooperation. In this region, the presence of chemosynthetic bacteria serves as a primary food source for larger organisms, showcasing how endosymbiotic partnerships enhance survival in inhospitable environments. The close association between these bacteria and their hosts facilitates nutrient exchange, enabling both to flourish in conditions that would otherwise be lethal. Corals present another fascinating case. These marine organisms house photosynthetic algae within their tissues, providing a clear example of mutualism. The algae converts sunlight into energy through photosynthesis, while the coral offers protection and essential nutrients. This relationship supports vast coral reef ecosystems, highlighting the vital role of endosymbiosis in biodiversity. In arid environments, certain fungi form symbiotic relationships with plant roots, allowing them to extract moisture and nutrients from the soil. This interaction exemplifies the adaptability of life, illustrating how endosymbiotic organisms can thrive in a variety of challenging habitats, underscoring the interconnectedness of all living systems. Each case emphasizes the significance of these cellular collaborations in the broader context of life's evolution. Understanding these cellular collaborations illuminates the intricate web of life that sustains ecosystems. Endosymbiotic theory suggests that the merging of different organisms led to significant advancements in cellular complexity. This principle extends beyond historical interpretations, influencing contemporary biodiversity conservation efforts. Endosymbiosis presents a framework for understanding how cooperation among different species can enhance resilience. In this region, mutualistic relationships are evident, where diverse organisms contribute to ecosystem stability. The presence of symbiotic partners often leads to the development of new traits, enhancing adaptability to environmental changes. Such adaptations are crucial in the face of climate change and habitat loss. Conservation strategies increasingly reflect these insights. By recognizing the interconnectedness of species, efforts to protect biodiversity take into account the dynamics of interactions within ecosystems. Preserving keystone species, which play a vital role in maintaining the structure of their communities, directly supports the overall health of these environments. Research into endosymbiotic relationships continues to reveal the depth of interdependence among organisms. These findings challenge traditional views of competition and survival, emphasizing the importance of cooperation in evolution. In turn, this understanding informs practices aimed at restoring degraded habitats, ensuring the survival of both individual species and the intricate networks they form. Thus, endosymbiotic theory remains a cornerstone in the ongoing quest to safeguard the planet's biodiversity. New technologies are reshaping the landscape of endosymbiotic research. Advanced genomic sequencing allows scientists to unravel the complexities of cellular interactions at an unprecedented scale. This capability unveils the genetic blueprints of symbiotic partners, offering insights into the evolutionary history and functional contributions of each organism involved. High-throughput sequencing techniques provide data that were once unimaginable, revealing the intricate relationships that define ecosystems. De imaging technologies, such as super-resolution microscopy, enable the observation of endosymbiotic events in real time. Through these observations, researchers can analyze how host cells integrate and regulate their symbiotic partners, shedding light on the mechanisms that promote or hinder cooperation. These advancements foster a deeper understanding of cellular communication and adaptation, essential for discerning the evolutionary dynamics of life on Earth. And Gall's ferritinumings are also being employed to engineer new symbiotic relationships. By manipulating genetic pathways, scientists can explore applications that may enhance agricultural productivity or develop biofuels through tailored microbial ecosystems. Research into artificial endosymbionts is expanding, offering the potential for innovative solutions to pressing global challenges. As this field progresses, a clearer picture emerges of the foundational role that symbiosis plays in shaping life. The future of endosymbiotic research promises to deepen our understanding of the interconnectedness of life and its implications for sustainability and ecological resilience. The interconnectedness of life reveals critical insights into climate resilience. Endosymbiotic relationships serve as a model for understanding how organisms adapt and thrive amidst environmental changes. The intricate partnerships formed between host cells and their symbionts illustrate the potential for cooperation and mutual benefit in nature. This concept extends to larger ecosystems where diverse species interact and adapt to shifting climates. Horniove Asmov. Certain organisms exhibit remarkable resilience through endosymbiotic arrangements. For instance, corals, which rely on photosynthetic algae for energy, showcase the importance of these relationships in maintaining their health and survival. Changes in temperature and ocean acidity threaten these delicate partnerships, impacting the broader marine ecosystem. Understanding the dynamics of endosymbiosis in this context highlights the potential for adaptability among various life forms. Research into endosymbiotic theory underscores a fundamental principle. Resilience is often rooted in relationships. It suggests that the capacity of species to endure environmental stresses may hinge on their ability to form beneficial partnerships. This insight fosters a broader appreciation for biodiversity and the intricate web of life that sustains ecosystems. As climate change intensifies, the application of endosymbiotic theory can inform conservation strategies and ecosystem management. Recognizing the significance of symbiotic relationships offers pathways to enhance ecological resilience, ensuring the survival of countless species in a rapidly changing world. The endosymbiotic theory presents a pivotal concept in the understanding of cellular evolution. It posits that certain organelles within eukaryotic cells originated as free-living prokaryotes, which entered into a mutually beneficial relationship with ancestral host cells. This transformative event laid the groundwork for the complexity seen in modern life forms. Mitochondria and chloroplasts exemplify this process, possessing their own DNA resembling that of bacteria, a testament to their independent origins. These organelles conduct vital functions, such as energy production and photosynthesis, which are essential for the survival of eukaryotic organisms. The integration of these prokaryotic cells into larger eukaryotic hosts marks a significant evolutionary leap, permitting the rise of multicellularity. The theory bridges microbiology with evolutionary biology, illustrating how cooperation and integration can lead to greater complexity and adaptability. This integration not only explains the origin of key cellular components, but also highlights the importance of evolutionary processes that shape biodiversity. Fossil evidence and genetic sequencing bolster the theory, revealing the intricate relationships and evolutionary pathways that have influenced life's development on Earth. These findings underscore the interconnectedness of species, emphasizing that cooperation can be as critical to survival as competition. The endosymbiotic theory invites a deeper appreciation of life's complexity and the subtle mechanisms that drive evolution. The legacy of endosymbiotic theory significantly influences contemporary biological sciences, reshaping our understanding of cellular evolution and the intricate relationships between organisms. It emphasizes the role of symbiosis in the evolutionary process, illustrating how cooperative interactions can lead to the emergence of new life forms. This framework extends beyond the origins of eukaryotic cells, offering insights into various biological phenomena, such as the development of complex ecosystems and the dynamics of microbial communities. These findings have led scientists to explore the importance of horizontal gene transfer, where genetic material is exchanged between different species, further blurring the lines of traditional evolutionary pathways. This understanding has profound implications for fields such as genetics, ecology, and evolutionary biology, prompting researchers to investigate the collaborative nature of life in diverse environments. Research on organelles like mitochondria and chloroplasts has established parallels in other organisms, reinforcing the idea that many cellular components share a common ancestry. Such connections underscore the unity of life and the shared evolutionary history among all living beings. This interconnectedness invites a reassessment of classification systems, urging scientists to consider evolutionary relationships that transcend conventional boundaries. As studies continue to unfold, the endosymbiotic theory remains a cornerstone of modern biology, illuminating the intricate web of life and the ongoing processes of adaptation and change. The enduring significance of the endosymbiotic theory lies in its capacity to elucidate the origins of complex life forms. This conceptual framework proposes that certain organelles within eukaryotic cells, such as mitochondria and chloroplasts, originated from free-living prokaryotes that entered into a symbiotic relationship. Such a merger provided distinct advantages, facilitating energy production and photosynthesis, which merges us. Research continues to reveal the implications of this theory beyond cellular biology, influencing fields such as ecology and genetics. The resulting energy efficiency led to larger and more complex life forms, fostering the development of multicellularity and, subsequently, the vast array of species present today. Insights derived from this theory have prompted further investigation into the genetic relationships among organisms, highlighting shared ancestry and evolutionary pathways. The concept also invites a broader understanding of cooperation in nature, illustrating how symbiotic relationships can drive evolution and adaptation. This perspective reshapes how biologists interpret interactions within ecosystems, emphasizing the importance of mutualism and co-evolution. By acknowledging these connections, scientists gain a more nuanced grasp of biodiversity and ecological resilience.